So today we're going to be looking at chapter 10, the muscle tissue. So there are three main types of muscle tissue. We've got skeletal, cardiac, and visceral or smooth muscle. This is also called smooth muscle. So looking at kind of comparing and contrasting what we see with these muscles. Obviously, skeletal muscles are going to be associated with the skeleton. That makes sense just based on the name. But the main function of these is going to be to pull on those bones to produce some sort of movement of the skeletal muscle. These things are going to be multinucleated. So that means we've got more than one nucleus per cell. And they're also going to be striated. Striated means it's like striped or lined, which you can actually see here with our striations that we see. Now, the interesting thing about skeletal muscle is it is going to be the only muscle that we're talking about that is going to be under voluntary control. You actually have to put some sort of conscious effort into causing it to contract or relax. This is going to be voluntary control. Cardiac muscle, on the other hand, is going to be associated with the heart, and it's responsible for generating the contraction and subsequent relaxation of the heart that's going to allow for it to pump blood. Now, these are going to look a little bit different. These are going to have only one nucleus per cell. However, they are still going to be striated with those stripes, just like what we see in our skeletal muscles. But a unique structure that we're going to see in our cardiac muscle is these things called intercalated discs, which you can see right here. These are these transverse thickenings that we have connecting these two individual cells to each other. We'll talk more about this in subsequent lectures. Now, obviously, cardiac muscle is going to be under involuntary control. You don't have to sit there and say, okay, beat, heart, beat, heart. You don't have to think about this. It is under involuntary control. And so it is under either autorhythmic control, so it has its own sort of pacemaker, as well as being influenced by different hormones and neurotransmitters that are going to all be responsible for the involuntary contraction and relaxation of this cardiac muscle. And then last but not least, we have our smooth muscle. We find this stuff all over the place. We find it in various organs, like the GI tract, for example, is going to be a great example of that. Functions vary. When we're talking about the GI tract, this is going to be a great example of peristalsis, right, where we have that kind of contraction and relaxation of the smooth muscles that line that GI tract that are going to help to squeeze foodstuffs through the intestines. These are going to have just the one nucleus, and this is a big difference, is they are not striated. These are very distinct looking when we compare them to the other two types of muscle. And just like what we saw with cardiac muscle, these are going to also be under involuntary control, and we have a certain degree of autorhythmicity where they are self-regulating this contraction and relaxation. We are going to spend the vast majority of our time for this chapter talking about the skeletal muscle. These other two will get covered in the cardiovascular chapters in subsequent lectures, as well as in the GI tract, the digestive tract later, in later chapters, essentially. So they've got their own chapters, so we're going to really mostly focus primarily on skeletal muscle. So, looking at the functions of our muscular tissue. Some of these are probably going to be pretty obvious, others might be something new. So for starters, muscular tissue is going to work to produce movement of the body. This can be movement of the whole body, like where you're moving your arms and your legs and your limbs all over the place, but it can also be this localized movement, right, where we have the contraction and relaxation of various kind of segments of the GI tract that help move foodstuffs through the system. All right, so it can be movement of the whole body or just very more localized movements. It's also going to help to stabilize body position. This is going to kind of come down to the skeletal muscle. These muscles are going to contract to stabilize joints, as well as to help to maintain your body's position. Things like sitting or standing helps keep everything going in the right direction. Muscles are also going to be important for storing and mobilizing various substances within the body. And this is going to be accomplished by this sort of ring-like band of smooth muscle that we call a sphincter, right? And this is going to prevent the outflow of various contents from your hollow organs. We've got sphincters all over the body, but this contraction that we have of our ring-like bands of our sphincters, right, keeps things from flowing out of the various organs. And then last but not least, a major sort of function of muscular tissue is going to be the generation of heat. So as muscle tissues contract, they are going to produce heat. And this process of heat production is known as thermogenesis. Thermo, like as in temperature, genesis, like creation of. So creation of heat, this is thermogenesis. This is why when you're going to the gym and you're exercising and you feel hot, this is because as those muscles contract, you're generating heat. This is also going to play a role in the involuntary contractions of skeletal muscles called shivering, right? This is going to help to, increase our rate of contraction to help produce heat when it's cold and when the environment is cold, to help warm you up. So generation of heat is a big deal. We then have some distinct properties that we associate with muscle tissue. For one, we've got electrical excitability. This is a really cool property. All right, this is basically essentially the ability to respond to various types of stimuli by producing electrical signals. All right, and these are known as action potentials, also sometimes called muscle impulses. I generally will refer to them as action potentials. But we've got two main types of stimuli that are going to trigger our action potentials, right? One is going to be the autorhythmic electrical signals that you have that are going to actually arise from the muscle itself. This is things like the heart's pacemaker within that sinoatrial node, right, where we're actively sparking up that stimuli on an autorhythmic sort of basis. The other option, though, is going to be chemical stimuli. This is going to be things like neurotransmitters that are being released from neurons or hormones that are going to be released from the endocrine system that are going to circulate through the blood and bind to their target cell. This can even be changes like local changes in pH. So all of these things are going to be chemical kind of classes of stimuli, though, that are going to ultimately result in in an electrical signal or an action potential being generated. Now, another property that we see with muscle tissue is contractility. All right, and so this is your ability for your muscle tissue to be able to contract forcefully, right? And this is going to be stimulated by our action potential. So an action potential is going to cause this contraction to occur. So when our skeletal muscles contract, it is going to generate tension, right? And this is going to be the amount of force of contraction that we have, this tension that's being generated by those skeletal muscles. And this is going to occur while it's pulling on the various attachment points, ultimately to try and generate some sort of movement, usually. And then last but not least, we have extensibility and elasticity. People get these mixed up a lot, so kind of bookmark this in your head for now. All right, so extensibility is the ability of that muscle tissue to stretch, all right, within limits without being damaged. So it has the potential to stretch. Elasticity, on the other hand, is that return to original length. This is going to be our snapback sort of capacity that we see in muscle tissue after we have either contraction or extension taking place. So extensibility is stretching. Elasticity is going to be our return to our original shape. So kind of make sure that you understand the differences there between those two terms, because those are easily and often, they're frequently mixed up. So like I said, we're going to spend most of our time looking at skeletal muscle for these lectures. So getting started on that. For starters, where are these muscle cells coming from? Well, muscle cells are called myocytes, all right? Myo meaning muscle, all right? Myo meaning muscle. And so each skeletal muscle fiber is going to arise during our embryonic development, so while you're cooking inside mom. And this is going to come about as a result of the fusion of hundreds of these little mesodermal cells called myoblasts. Now, once these myoblasts fuse, right, they're going to become these mature skeletal muscle fibers. And this is why we see these multinucleated sort of structure that we've got going on here with multiple nuclei, because we've got the fusion of multiple of our different little myoblasts here. But once fusion occurs, our muscle fibers have now lost the ability to undergo cell division. So you're kind of stuck with the number that you've got. So the number of skeletal muscles that you have is pretty much set before you're even born. Right? And hopefully most of these cells are going to last your lifetime. That being said, when you're going to the gym and getting all buff and swollen, whatever, you're not increasing the number of skeletal muscles that you have in your body. You're simply increasing the size. So that's going to be how you go in and have your muscle gains or whatever people call it these days. All right? So moving forward then to look at the connective tissue that we see associated with skeletal muscle. For starters, of course, we've got our subcutaneous layer. We've mentioned this structure, this layer before. This is just deep to the dermis. And this is what's going to separate the skin from the actual muscle. Right? This is, of course, made up of areolar connective tissue as well as of adipose tissue. This is a fantastic sort of passageway for things like nerves, the various vessels that you have, lymphatic, blood vessels, etc. That are going to be associated with the muscle, and this is going to be providing their entry and exit point for those muscles. Now, the adipose tissue that we have associated with our subcutaneous layer is also going to be fantastic for the storage of triglycerides, of our body's sort of long-term fat storage, energy storage there. And this is going to serve also as an insulating sort of layer that's going to help to reduce any kind of heat loss. And it's also going to provide us a nice little bit of cushion, protecting the actual muscle from any sort of physical trauma. All right. So then moving on to the rest of the connective tissue that we have associated with the muscle tissue. Right. First of all, we have our epimysium. All right. Epi means on top of. Okay. So this is going to be our outer layer. This is going to encircle the entire muscle all the way around is going to be the epimysium. Right. And this is going to be made up of a dense, irregular connective tissue. Perimysium next, peri meaning beside or, with, is going to be our layer of dense, irregular connective tissue. And this is going to surround these groups of anywhere from 10 to 100 of these little muscle fibers. You can see here, these little bundles are known as fascicles. All right. So the perimysium is going to surround the actual fascicle itself. The endomysium, endo like within, inside, all right, is going to penetrate into the interior of each fascicle, and it's going to separate these individual little muscle fibers from one another, as you can see right there. And this is going to mostly be made up of reticular fibers. Okay, so that's kind of our breakdown of how we have our arrangement of our connective tissue. There's also going to be stuff called fascia. And these are these sheets or either bands of fibrous connective tissue that are going to be deep to the skin, and they're going to surround that muscle and other organs that we see with the body. And so if the connective tissue extends beyond the muscle, right, to form kind of a rope-like structure, then we call that a tendon, which we can see here. All right, it's a tendon. If it forms a flat sheet, then it's going to be called an aponeurosis. We have this on top of your noggin. Kind of connecting the occipitalis and the frontalis muscles. It's connected by an aponeurosis, a flat sheet of connective tissue. So then looking at the microscopic anatomy that we see with muscle, right? For starters, just so that we're aware, people who study these things, they had to come up with different names because they couldn't just go with the normal cell names. They had to get fancy. So the sarcolemma is going to be the plasma membrane for a muscle cell specifically. All right, so that's just the plasma membrane in our muscle cells. Likewise, the cytoplasm itself for our muscle cells, this is known as the sarcoplasm. So the sarcolemma is the plasma membrane, sarcoplasm is our cytoplasm, and is typically going to contain large amounts of glycogen, fantastic for energy production. We're also going to see a good bit of myoglobin stored within the sarcoplasm. This is just going to be another oxygen-binding molecule, kind of similar to hemoglobin. Another structure that we're going to see within the muscle is going to be the transverse tubules, also known as T-tubules. These are these little invaginations that you can see here. They're kind of blue, looking like that, right? Those are our T-tubules. These are going to be these sort of dips in the sarcolemma. And this is where we're going to have the ability for our muscle action potentials to be able to spread very quickly throughout all parts of that muscle fiber. Now, our myofibrils are going to be these little thread-like structures that we have within the sarcolemma. And these are going to be our contractile structures that we have, our contractile organelles within the skeletal muscle. These are the ones that are doing all the work when it comes time for that muscle to contract. We then have our sarcoplasmic reticulum, which is very similar to what we see for the endoplasmic reticulum in a normal cell. This is going to actually encircle this is going to encircle the myofibril. This is this kind of like, I don't know, kind of weird, maybe tan color things that we've got going on here. These guys right here, these are our sarcoplasmic reticulum. But this is going to be fantastic for the storage of calcium. All right. The terminal cistern, on the other hand, is just going to be the dilated sac portion of our sarcoplasmic reticulum. And this is going to butt up against our T-tubule. So we've got kind of a sandwich on either side. And this sandwich of our terminal cistern, right? We've got our T-tubule and then another terminal cistern, right? This whole group here is then going to be called a triad, all right? Now, this structure that we have is really super important when it comes time for the muscle to actually contract. So when our muscles are relaxed, the sarcoplasmic reticulum is going to be responsible for storing those calcium ions. However, when we're ready for contraction to be initiated, we're going to release calcium from that terminal cistern of our sarcoplasmic reticulum, and this is what's ultimately going to trigger the contraction of this muscle. So kind of bookmark that for later, because it all comes down to this release of calcium that's going to sort of trigger our muscle contraction. So looking at the different filaments that we have making up the individual, these myofibrils, all right? So within the myofibrils, of course, we have these smaller proteins that are known as filaments. We have two main types of filaments. We've got our thick filaments and our thin filaments. Now, our thin filaments are going to be made up of a protein called actin. Thick filaments are going to be made up of the protein myosin. All right? Now, both types of filaments are going to play a role in the contractile process. They're going to work together. There are going to be two thin filaments for every one thick filament, and they're going to form this sort of region of filamental overlap. So if we've got our thick filament, right, and we've got our thin filament, there's going to be this region of overlap where we've got kind of an overlapping sort of region here. That's going to be our filamental overlap. And that's going to be crucial when it comes time for actually contraction of these muscles, of these myofibrils. Now, the filaments that we see inside of our myofibrils, they are not going to extend the entire length of our muscle fiber. Instead, they're going to be arranged in these functional little compartments called sarcomeres. And this is the basic functional unit that we see for our myofibril. All right, so here we can kind of get an idea of what that's going to look like. All right, so here's what we're going to do. We're going to go through and we're going to look at all the components of our sarcomere. And then on this page, there is an image of the whole thing. What I want you to do after you listen to this lecture is I want you to pull these two slides up side by side and say, okay, let's look at what these structures are and then let's compare them to what I see for the actual images. So just keep that in mind when we're going through it. So looking at the components, all right, we arrange these things into bands and zones, all right? Now, the narrow kind of plate-shaped region that we have that's made up of these dense protein material is called the Z-discs, all right? And this is going to be the borders of our sarcomere. So our sarcomere is going to extend from Z-disc, all the way to Z-disc, and that is going to be, the whole thing is going to be our sarcomere. All right, so those are the borders of our sarcomere. We're going to have a dark middle portion of the sarcomere that's called the A band. So there's an A in dark and there's an A in A band. Oh my heavens, I'm so sorry. And this is going to extend the entire length of the thick filament. All right. Now, towards the end of each of these A bands, there's going to be our zone of overlap, right, where we're going to have both thick and thin filaments. They're going to be lying side by side, okay? Remember like what we drew just a second ago, okay? So we've got this kind of zone of overlap here, all right? This right here is our zone of overlap. And then our I band is going to be lighter. So there's an I and I band, there's an I and lighter. All right. This is going to be a less dense area that is going to contain all the rest of those thin filaments, but it's not going to have any thick filaments there. And our Z disc is going to pass smack dab through the middle of each I band. All right. So in the middle of our Z band, there's going to be an I band. All right. Now, we're going to have this alternating sort of pattern of I bands and A bands. So this dark A band, light I bands. And this is what's giving us that sort of striated pattern that we're seeing within those myofibrils. And this is going to extend the whole length of this pattern is going to be the whole length of our myofibril. And we see this in skeletal and cardiac muscles. And it's actually visible under a light microscope. It's pretty cool. We're also going to have this narrow little H zone in the center of each A band, and this is going to be where we're only going to have our thick filaments, no thin filaments, all right? And then supporting proteins, of course, are going to help to hold all of these things together. The thick filaments are going to be held together in the center of our H zone. This is going to form the M line, which is named M line because it is in the middle of the sarcomere. Very creative naming system there. Right? So looking at what we've got here. Here we've got our M line, right? This is our M line smack dab in the middle of our sarcomere. Our sarcomere is extending from Z disc to Z disc with everything in between being our sarcomere, okay? We've got our zone of overlap right here. All right? We've got our, what are we looking at here? We've got our, I should use a different color probably. We've got our A bands, our dark zone here, right, that we can see. And then we've got our zone of overlap. We've got our I bands that are going to pass through that Z band there, that Z disc, excuse me. And then the A band, I band, I think I got them all. All right, with our Z discs there kind of making up the middle. And then our M line smack dab in the middle. All right, so that's the kind of arrangement that we're seeing with our sarcomere. So this is how it looks microscopically if we were to look at this thing. And we are able to see these striations under just a normal sort of like microscope. So that's really pretty cool. We can also zoom in a little bit. Oops, gosh darn it. Zoom in a little bit and we can see here where we've got, okay, this is the boundary of our sarcomere here. We've got our zone of overlap where we have both thick and thin filaments with both actin and myosin. We've got our I band where we've just got our light thin filaments there, and then our A band where we've got thin and thick filaments, okay, as well as the what am I missing? I think that's it. Oh, excuse me, our H zone. Here we go, our H zone, which is just going to have those thick filaments. All right. So on to the, carrying on with the components of our sarcomere. How it actually looks when we look at it under a microscope. This is using transmission electron microscopy. So this is Mac Daddy magnification. We're looking at 21,600 times magnification in order to be able to see this. But it's really pretty cool. All right. So of course here we've got our Z discs, forming the bounds of our sarcomere. All right. We've got our I band, which is going to be light fibers only. We've got our dark A band, which is going to contain our zone of overlap, which we see here, okay, which is going to have our H zone, which is just thick only, our zone of overlap, which of course is going to have thick and thin, and then our I band, which is going to span the Z disc, which is going to be light only. All right, so pretty cool, huh? And then there's our M line down, smack dab down the middle. So, there you have it. Those are the components of the sarcomere. This, I'm telling you, all of these components are important, all right? They are a big deal, all right? You're going to need to know them. So looking at the various proteins that we have making up these muscles, we group these different proteins into three main kind of categories, right? We've got our contractile proteins. These are the ones that are actually responsible for generating our force of contraction when we're ready to contract. We've got regulatory proteins that are there to sort of make sure that we're switching contractions on and off and are going to sort of regulate that process, hence the name regulatory proteins. They're going to control whether or not we're going to actually contract, all right? And then we have our structural proteins. These are there to sort of keep everything in proper alignment. They're going to keep everything lined up properly. They're going to provide us with the distinct characteristics that we see for our muscle cell tissues, with our elasticity and extensibility. And they're also going to link our little myofibrils to the sarcolemma as well as to the extracellular matrix. So we're going to look at these, starting, I'm pretty sure, with the contractile proteins. So these are pretty cool. So we've got two main contractile proteins. We've got myosin and we've got actin. Now, myosin is going to be the major player in the thick filaments. And this is going to be our motor protein in all three types of muscle tissue. Now, our motor protein are going to pull, all right, on various cellular components, and they're doing this in order to achieve movement. But they have to have energy for that, and so they're going to rely on the conversion of hydrolysis of ATP to convert that energy, that stored energy, into the actual mechanical energy that we need for motion. Right? This is what's ultimately going to generate our force of contraction, is this conversion of ATP. This is going to power this whole cycle. Now in skeletal muscles, we've got about 300 or so molecules of myosin that are going to make up every single thick filament, which we can see here. There's our thick filament, all right? And each little myosin molecule is going to be looking like two golf balls that we've twisted their little tails together, all right? So the myosin tail, which is like the golf club handle part, all right, is going to point towards the M line. We're pointing towards the M line. The M line's right there, all right? In the center of our sarcomere, right? And the tails of our neighboring myosin molecules are going to all lie like nice and lined up parallel to each other. Isn't that nice? All right. This is going to form the shaft of our thick filament. Now, coming up off of each of these little myosin molecules are going to be these two sort of projections that we've got going on, all right? This is going to be kind of like the golf club head in this sort of instance here. And these are actually called the myosin heads, all right? Now, each myosin head is going to have two binding sites, and this is important, all right? We're going to have a binding site for actin where it's going to bind to that other motor protein, okay? And then we're going to have a binding site for ATP. And this is also going to have an enzyme sort of functionality to it. It's going to work as an ATPase, right? So this is going to be an enzyme that's going to hydrolyze ATP, converting it from ADP, converting it from ATP into ADP, plus our little phosphate molecule. This is going to produce our energy, right? This is going to be our energy reaction. So ATP is going to be able to bind here, and it's going to have this hydrolysis reaction, taking place at this binding site to generate the energy that we need for our muscle contraction. Now, the heads that we see on our myosin fibers here are going to project outwards from that shaft in sort of spiraling kind of arrangement, and are each going to extend towards one of the 6 thin filaments that are going to surround each of our little filaments here. All right. So this is myosin. This is going to be our thick major player in our thick filament. All right. Actin is going to be our other contractile protein. This is going to be the main component of our thin filaments. Right? Now, each little actin molecule that we can see here, these are all individual actin molecules. These are going to be joined together to form these actin filaments that are going to have a twisting kind of double helix structure to them, kind of like what we see in DNA, where we've got this sort of twisting situation going on. Pretty cool looking. And each actin molecule is going to have a myosin binding site where that myosin head is going to be able to attach, right? So here we've got our actin binding site, and this right here is going to activate, and it is going to bind to the binding site, to the myosin binding site that we have on our actin. So they're kind of matching up. It's great. We're going to form a crosslink here. So we've also got 2 major regulatory proteins that are associated with this, all right? This is going to determine whether or not we're turning on or off our contraction cycle. Our 2 regulatory proteins, are going to be tropomyosin and troponin. Now, when our muscles are relaxed, these little myosin binding sites, they're blocked from being exposed, to being able to bind to myosin, right? On that actin. So they're going to be covered by this protein that we call tropomyosin. Good grief, I'm sorry. All right. And then troponin, which we can see here, this is troponin, all right, is going to hold our tropomyosin in place so that it is actively blocking our myosin site. So it can't have any interaction. It's not allowing it to happen. No binding between myosin and actin at this point. Right? However, when we have calcium levels, right, when we have an increase in intracellular calcium in our myofibril, this calcium is going to bind to our troponin, and it is going to pull it out of the way. Right? When this conformational change is happening, when our shape change takes place, it's going to pull our tropomyosin out of the way. This is going to expose our myosin binding site that we have on that actin, and this allows for those myosin heads to now be able to bind to the actin site, the actin molecules, right, allowing for a crosslink to form. Right? This is super important. It's this binding between myosin and actin as a result of tropomyosin being pulled away because of this shape change that we see in troponin, exposing that myosin binding site. So last but not least, our final sort of category that we have for our different proteins that are in our myofibrils is going to be the structural proteins. These are going to play an important role. They're super critical, but they're not directly going to necessarily be involved in the actual contraction cycle or the contraction process. All right. These are going to do a wide variety of things. They're going to keep things nice and aligned and organized with the thick filaments and the thin filaments so that we can have that binding happening when we're ready for it. This is also going to give us the elasticity and extensibility that we see as a structural kind of characteristic of the different muscles. And it's also going to help to link our myofibrils to the actual sarcolemma and the extracellular matrix. So starting with titin. This is going to be a structural protein that's going to connect Z discs to the M line. Titin is I'm pretty sure titin is one of the largest proteins that you have in your body. If you ever meet a scientist who studies titin, you should feel very bad for them because everything is more difficult because it is so darn huge, which is where it gets its name. So ultimately, titin is going to help with stabilization of our thick filament position. Right? It's going to help our sarcomere return to the resting length after our muscle has contracted or been stretched. We also then have alpha-actinin. All right, this is another structural protein that's going to be part of the Z disc, and this is going to bind actin molecules of the thin filaments to the titin molecules that we saw above. Myomesin over here is going to be another structural protein. This is going to make up the M line. This is going to form our M line of our sarcomeres, and it's going to help to bind titin molecules and connect adjacent thin filaments to each other. Nebulin is another structural protein, of course, and this is going to wrap around the entire length of each of these thin filaments. And this is going to play a role in keeping the alignment of our thin filaments within the sarcomere, keeping them lined up properly. And then last but not least, we have dystrophin, all right? This is going to be our last structural protein here. This is going to link our thin filaments of our sarcomere to the various integral membrane proteins within the sarcolemma. All right. It's also going to help to reinforce the sarcolemma, and this is critical because this is going to help to transmit the tension that's generated from the sarcomere to those tendons. So it's helping to sort of shift the tension that's being generated to other kind of parts of the muscle, to the actual connective tissue tendons. So here, in case, for your studying delights, I've included a table summarizing all of the various proteins that we have in our skeletal muscle fibers. Very exciting stuff. So now that we've got all the players, let's look at how it works. So looking at the contraction and relaxation process that we see for our skeletal muscle fibers. This is going to rely on something called a sliding filament mechanism. All right, this is where muscle contraction is going to occur because our myosin heads are going to sort of are going to attach to and walk along the thin filaments at both ends of our sarcomere, pulling progressively those thin filaments, pulling them towards the M line. All right, this is going to cause that shortening that we see of our muscle. So myosin is going to pull on actin, and this is going to cause those thin filaments to slide inward. Okay, now as a result of this, the Z disc is going to move towards each other. Again, we're having this sort of, as we contract, look at where these Z discs are here when we're relaxed versus where our Z discs are here when we're fully contracted. See, they've slid inwards towards the midline, okay? Now, all of this is, as a result of our various structural proteins, this is we're going to allow for the transmission of force that's going to be spread through the entire muscle. And this is going to result in the whole muscle contracting, not just a little individual bit of it. And so you can see here, look at the changes, the shift that we've seen in our eye band. Look at how big this was before, right? Our eye band is here. And it's starting to shrink, and then it's even smaller here. This is our eye band, all right? Again, also look at our zone of overlap that we can see here, our zone of overlap, all right? At fully relaxed versus at fully contracted, this is now our zone of overlap, our ZO, all right? As opposed to teeny tiny right here, much smaller, So you again can see how this sliding filament mechanism is causing this this shortening of our muscle, all right, with this sliding filament mechanism. Now, this brings us then to the contraction cycle. So we only have 4 easy steps to go with this. First of all, we have ATP hydrolysis. So we're going to have ATP. It's going to, is going to bind to that ATP site, right? And it's going to get converted into ADP plus our little phosphate molecule, which we can see here. All right. Now, this is important because with this ATP hydrolysis, this is going to energize and orientate our little myosin head here now. So we are now at the appropriate angle. Everything's lined up and ready to go so that we're ready to bind to actin. So once that's happened, we're now going to then have attachment where our myosin head is going to actually bind to actin. And this is going to form this cross bridge here that we have, this connection that we have in between the myosin and our actin filaments. And then we're going to have our power stroke. This is where we're going to pivot and pull, all right? This is where we're going to have, this change in direction, and we're pulling those Z discs towards the M line, right? The thick filaments are going to pull those thin filaments past our thick filaments, and pulling them towards the center of the sarcomere. Now, once this happens, though, ADP is going to be released. It's like, yeah, we're done here. I'm leaving. Peace out. All right, so ADP leaves. This is going to cause detachment to take place, where myosin is going to bind ATP. This is going to another molecule of ATP, right? We're going to have the detachment of our cross bridge. So myosin is being released from actin, and then we're ready to start the whole situation all over again, right? Where we have ATP is now going to be hydrolyzed to ADP plus our phosphate molecule, which is where we are here. All right, where we're now energized and orientated. We're ready to have the cross bridge forming, where we're having this attachment between myosin and actin. All right. We then are going to pivot and pull. Okay, that myosin head is going to pull that thin filament past its thick filament. Okay, and then this is going to cause the release of our ADP. That's done. We bind a new molecule of ATP that causes the detachment between myosin and actin, and we're ready to start the whole process over again, where we have hydrolysis of ATP once again. So that right there is the contraction cycle. This is important. Yeah. So I won't show you now, but you're welcome to click on this link later if you want. This is just a little motor protein, sort of, you can see it walking along those microtubules doing that contraction cycle. It's fun to look at. It's got funny music, just in case you're interested. So let's put this together now, because we've now seen how it's going to pull on that to have that contraction happening, but what's triggering that? This is where we have to have this excitation contraction coupling, right? Where we're having some sort of excitatory influence that's going to trigger our contraction and putting these two things together. So we've mentioned calcium a couple of times now. So during relaxation, the levels of calcium in the sarcoplasmic reticulum, right, so in the actual cell, are going to be fairly low, all right? This is because within our sarcoplasmic reticulum, we're actively pumping calcium into the sarcoplasmic reticulum. This is keeping our levels of calcium high in the sarcoplasmic reticulum. Now, when our little muscle action potential, lightning bolt, action potential, right? When this propagates and spreads along our transverse tubules, all right? It's going to spread through that whole thing. This is going to cause the opening of our voltage-gated calcium channel, okay? And this is going to allow for calcium to spill out into the sarcoplasm. So we now are going to have an increase in intracellular calcium within that sarcoplasm, all right? Calcium is then going to be able to bind to troponin. It's going to pull that tropomyosin out of the way. This is going to expose those binding sites and allow for that cross bridge to form, all right? This is a super critical concept because this is going to connect that muscle action potential, connects it to our sliding filament mechanism and the actual contraction cycle altogether. So this is a really critical kind of idea to sort of understand how we're bringing it all together, how when we have our action potential is going to spread through our transverse tubules, and this is causing the opening of these voltage-gated calcium channels, causing this calcium to leak out into the sarcoplasm, where we're now going to have a high intracellular calcium that's going to allow for this contraction cycle to proceed. All right. Now, I'm just going to summarize this for you because it's a little bit over the top and I need to change it, but I always forget. So, looking at this relationship that we see between length and the amount of tension that can be generated. This is essentially a Goldilocks situation here. Too much stretch, right? You stretch beyond 170 or so percent of our, normal sarcomere length. We're going to lose our zone of overlap, and we're going to be we're not going to have enough zone of overlap for that contraction to be able to pull towards the midline. So this is not going to happen. All right. Same deal if we have we shrink the shorten the length of our resting sarcomere. All right. When these things are too short, right? This is going to cause the alignment that we see of our zone of overlap to be out of alignment. So we're not going to be able to generate any sort of contraction. If you've ever done like a big heavy squat and you get stuck at the bottom of the squat and you can't quite stand back up, this is probably because you've overstretched, those filaments, right? So again, it's kind of a Goldilocks zone. We're pretty flexible here, but anywhere between 80 and 120% of the length of our sarcomere is going to be sort of an optimal kind of range, with this, being the primary thing. It's going to be able to be achieved, right? So this is just to say that normally, our muscle resting muscle fiber length is going to be held pretty close to this range, so that we're able to, initiate a contraction whenever we're ready. All right. So now that brings us then to this idea of our neuromuscular junction. This is an important idea. All right. So a somatic motor neuron. This is going to be a neuron that is going to stimulate skeletal muscle fibers. It's going to cause them to contract. If it doesn't stimulate our skeletal muscle fibers, then it's not a somatic motor neuron. This is specific to our skeletal muscle fibers. All right. If it doesn't, work with the skeletal muscles, it's some other kind of motor neuron. All right. So the muscle fibers are going to contract in response to action potentials. To one or more action potentials, they're going to propagate and spread along that sarcolemma through our system of T tubules, allowing this to spread nice and easy and spread throughout the whole myofibril. Now, the muscle action potential is going to arise at our neuromuscular junction. This is going to be the synapse that we see between our somatic motor neuron and our skeletal muscle fiber, right? So we've got that's my neuron there. This is going to be our somatic motor neuron, okay? And on the opposite side here, we've got our skeletal muscle, which I am not great at drawing. All right, so this is going to be here where this whole thing is coming together. This is our skeletal muscle. Bear with me. All right, this whole thing here is going to be the neuromuscular junction. All right, now a synapse is going to be a region where we're going to have communication occurring between either between two neurons or between a neuron and a target cell, which is what we're talking about in this case, right? So in the case of our neuromuscular junction, this is going to be between our somatic motor neuron and our skeletal muscle. These are going to be where we're going to form our synapse. Now, at most synapses, there's going to be this little gap, all right? We call this a synaptic cleft, all right? This is going to actually physically separate those two cells. Now, that being said, because these two cells don't actually physically touch, our little action potential that's propagating down our somatic motor neuron isn't going to be able to jump this gap here, our synaptic cleft, and it's not going to be able to spark up, our action potential. So how do we get around this? Instead of trying to jump the gap, what we're going to rely on is for the release of these various chemicals, right, that are going to allow the communication between these two cells. All right, these chemicals are known as chemical messengers, and in this case, we call them neurotransmitters. Now, at the neuromuscular junction, at the end of our motor neuron, this is going to be where we're going to have our axon terminals, right? And these are going to be divided into little clusters of our synaptic end bulbs. This is going to be the neural portion of our neuromuscular junction. This is the N, right? Now, suspended within the cytosol of each of our little synaptic end bulbs are going to be hundreds of these membrane-enclosed sacs that are called synaptic vesicles, right? And within our synaptic vesicles, they're going to be absolutely packed full of neurotransmitters, all right? Now, the neurotransmitter of choice for our somatic motor neurons is going to be acetylcholine. Right? This is going to be the neurotransmitter that gets released at our neuromuscular junction. Now, the region of the sarcolemma that is just going to be opposite our synaptic end bulb is called the motor end plate. And this is the muscular portion. This is the muscular part of our NMJ, all right, of our neuromuscular junction. So within each motor end plate, there are going to be millions and millions of receptors, specifically acetylcholine receptors, right? So when we have the release of our acetylcholine, right, it's going to be able to diffuse across the synaptic cleft, and it is going to be able to bind to the receptor on that motor end plate, okay? These receptors are going to be packed in tight, and we're going to make use of something called junctional folds, which are these deep grooves in our motor end plate. This is going to allow us to have an increase in the surface area that's available. This allows us to pack in more of these little receptors for acetylcholine so that we can maximize the number of these little receptors, so that we can maximize the amount of neurotransmitter that we're able to bind to, specifically in this case, again, acetylcholine. So our neuromuscular junction, as a result of this, is going to include our synaptic end bulb, which we're going to draw kind of like that. Bear with me here on one side. We've got our synaptic cleft, okay? We've got the actual synaptic cleft itself, obviously. And then we have our motor end plate, which I struggle to draw, I'm not going to lie. There's our motor end plate. And this whole thing right here is going to make up our neuromuscular junction, all right? So we've got our synaptic end bulb, and our motor end plate, all right? This whole thing is going to make up our neuromuscular junction. So the way that this works, all right, what's happening at our neuromuscular junction that's allowing us to spark up this action potential, all right? So for starters, we've got our action potential that's going to propagate down our somatic motor neuron. It's going to trigger the release of our neurotransmitter. So what's going to happen is our action potential is going to travel along our the axon of our somatic motor neuron, and it's going to trigger the opening of our voltage-gated calcium channels. All right? Oh, gosh darn it. We're now going to have an increase in our intracellular calcium, okay? And that is going to trigger exocytosis of our synaptic vesicles. All right, so we're like, release the neurotransmitter, and we're going to release all of our little acetylcholine, right? And it's going to diffuse across our synaptic cleft, and it is going to bind to receptors on that motor end plate. Now, once we have our acetylcholine binding, right, it is going to open our ligand-gated or our acetylcholine-gated channels, right, and is going to allow for sodium, positively charged sodium, to enter into the cell. So with this increase in our intracellular sodium, this is going to cause a depolarization, which is ultimately going to spark up our action potential. All right. Now, what about when we're done? We're like, okay, we've had enough. We're done with the binding. What we're going to do is we're going to call in an enzyme called acetylcholinesterase, and it's going to come in and it's going to go chop, 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 and it's going to break down all of these little extra acetylcholine molecules so that we don't get any more binding, and this is going to ultimately shut our ligand-gated sodium channels, allowing for a drop in our intracellular sodium. All right, so that's what's happening. So looking at this, putting it all together, this is where I love this image. I think that it is fantastic because it just literally sums up the entire lecture that we had just in one great picture. So what's going to happen is we've got our action potential. It's going to propagate along the axon of our somatic motor neuron. It's going to get to our axon terminal here, and this is going to cause the opening, all right, of our voltage-gated calcium channels, okay? This is going to result in an increase in calcium, all right, which is going to trigger the release, the exocytosis of our synaptic vesicles. So we're releasing our acetylcholine into the synaptic cleft. This is going to bind to our little receptors, all right, that we have, all right, and this is going to open up our acetylcholine-gated sodium channel. So now we have sodium flowing into our cell, all right. We've got an increase. This is going to cause that depolarization. That's going to spark up our action potential, which is going to propagate along our cell, all right, along our myofibril, through our transverse tubules, spreading throughout the cell, all right? With the spread of our little action potential here, this is going to open up our voltage-gated calcium channels, all right? Causing calcium to leak out of our sarcoplasmic reticulum, all right? Leaking out, and we're now having an increase in our calcium levels that we have in the sarcoplasm, all right? Now, with this increase in calcium, this is going to bind to troponin, and that's going to pull tropomyosin out of the way, exposing, right, we've bound, our calcium right there is bound to troponin, pulling tropomyosin out of the way, exposing the little myosin binding sites that we have on our actin filaments. We're then going to have our contraction cycle happening, right, where we have myosin binding to actin, right, as well as a molecule of ATP, right? We're going to have that cross bridge formation taking place. We're going to have our power stroke where we're going to pivot and pull. Then we're going to release, and that is going to cause our thin filaments to slide towards the center of our sarcomere, right, towards our M line. Now, when we're done and we're ready to relax, all right, at that point then, we're going to call in the calcium ATPase, right? And we're actively going to spend energy. We're actively going to convert ATP, right, into ADP, plus our little phosphate. We're spending cellular energy to pump this calcium, which is high outside in the sarcoplasm, right? All right, we're pumping it back into our sarcoplasm, and this is going to cause the drop in our calcium levels in that sarcolemma. With the removal of these high levels of calcium, this is going to allow for our troponin to return back to its original shape. Tropomyosin is going to slide back over and cover those little myosin binding sites, and our whole muscle is going to relax. And that right there is the whole situation of excitation, of contraction, and relaxation of our skeletal muscle, all in a nutshell, all in one fabulous, wonderful little picture. All right. This is a fantastic picture. I love it. I highly recommend you take a look. So With that, that is the last that I have for this portion of the lecture for our skeletal muscle. So I will see you all next time. As usual, please feel free to get in touch if you have any questions, comments, or concerns. See you next time.